Now that we've looked at filtration, let's look at the other two processes that occurred through the nephron, reabsorption and secretion. Let's talk about reabsorption first. Now, if you can remember first semester, you'll remember diffusion, ion channels, channel-mediated and carrier-mediated facilitated diffusion, active transport and osmosis. All these processes are used in reabsorption. We've also got a couple of new processes. Facultative water reabsorption and travelling between cells. Now, the cells in the proximal convoluted tubule, or PCT, actually have gaps between them. Very small gaps, but they're big enough for ions and water to come through. Facultative water reabsorption means that it requires ADH, or antidiuretic hormone, something we'll talk about further on. This whole process we will talk about further on, but it means that the water reabsorption does not happen through osmosis, but with help from ion channels and hormones. Reabsorption, most importantly, happens from inside the tubule, across the tubule wall, across the interstitium, through the capillary wall and into the blood. So it is quite a complicated process. Over here is a summary of the processes that go through the nephron. So we've had a look at filtration, and now we're having a look at reabsorption, which goes back into these peritubular capillaries you'll remember from your anatomy. And that will go through to the renal vein, and what's left will be excreted. So reabsorption, where does it happen? Well, we've said it happens in the nephron, yes. But more specifically, most of it happens in the proximal convoluted tubule. This is the primary site for reabsorption. 60 to 70% of sodium is reabsorbed, as is 60 to 70% of water. Mostly this happens through passive methods, so no energy is required. But there is part of it that requires energy. Glucose, amino acids and bicarbonate are all reabsorbed here. Glucose is of course the energy molecule in our bodies, and with diabetes it overruns the channels that exist to reabsorb the glucose, and it comes out in our urine and can be measured. Amino acids are the building blocks of proteins, and these generally have the same amount of ion channels as there are amino acids, so we don't have many of them in the urine. And bicarbonate, we'll talk about a bit further on. It's something called a pH buffer. It is used to look after pH and keep the number of hydrogen ions within range that is best for our body. The loop of Henle is also important for sodium and water reabsorption. You'll notice that there's a bit more sodium reabsorbed than water, and we'll talk about that further on. The distal convoluted tubule, reabsorption is still going on, however now it is less so. It is predominantly sodium that is being reabsorbed, and most notably it is relatively impermeable to water, except with the presence of ADH, or antidiuretic hormone. Then the collecting duct. We are still reabsorbing at the collecting duct a bit of sodium and a bit of potassium, the two main ions in our fluids. So that's a brief look at reabsorption. It takes a lot of different methods to get substances from the filtrate back into the blood. Now we'll look at secretion. Secretion is about removing substances from the blood into the tubule that haven't been filtered, or that have been filtered reabsorbed, and then secreted. They go through quite a process to be excreted, which means put out in the urine. Important substances that are secreted are potassium and hydrogen. Hydrogen, of course, being pH, and this requires active transport, so it takes energy to do this. The wastes ammonium, creatinine and urea are also secreted. These are sometimes reabsorbed and then secreted again. Some larger molecules get put out this way. They're not able to fit through the filtrate system, so some hormones, including human chorionic. Some larger molecules are put out this way as well. They're not able to be filtered. But some hormones, including human chorionic gonadotrophin or the pregnancy test hormone, are secreted this way. Foreign substances, such as some drugs, penicillin, are secreted. And this occurs from the loop of Henle to the collecting ducts. So not in the proximal collecting tubule, but everywhere else. Now remember, this time it goes from the blood supply, which will be the vasa recta or peritubular capillaries, through the extracellular space, through the tubule wall, into the filtrate, and ends up in the filtrate from the blood supply. So it's going backwards really, and as you can see, go through many steps again. Now let's have a chat about acid-base balance. Just to remind you, pH is the concentration of hydrogen ions. It affects protein shape, chemical reactions involving hydrogen, of which there are many, and general cellular function. So pH is quite important to keep balanced. Kidneys play a huge role in balancing acid-base and are the only way of removing many acids. So we talked about how in the lungs we blow off CO2 and that takes out carbonic acid. Most other acids are removed from the kidneys and they're excreted or put out in the urine. However, there is also a buffer system. What is a buffer system? Let's have a look. So buffers are molecules that are present in the intracellular fluid, extracellular fluid and the urine, and what they actually do is bind with hydrogen ion to take it out of circulation. So they undergo a chemical reaction and form a new molecule or ion that it doesn't allow the hydrogen to have such an impact. However, the important part of buffers is when the pH gets too high, so it becomes too alkaline, they release hydrogens again to lower pH. So the hydrogens are captured in the buffer, but can be released when needed. Some examples of proteins. These are a very important buffer. Phosphates, these are mostly found in the cell. And bicarbonate and ammonia. Now, ammonia and bicarbonate. Ammonium and ammonia are the same except for a hydrogen ion. Bind with hydrogen in the urine. So one thing they do is take the hydrogens out of the urine so that more can be secreted in. They're also produced by the kidneys, so the kidney cells produce ammonium and bicarbonate, which goes back into the blood. And then they also reabsorb the ammonium and bicarbonate that has been filtered, and so it goes back into the blood where it can work to manage the pH level. Now, speaking of the pH level, these three processes are controlled by the filtrate concentration of themselves and of hydrogen, blood concentration of themselves and of hydrogen, so the pH, and hormones which look after the pH level.